Good afternoon, everybody, and a big welcome to this British Pharmacological Society webinar on the therapeutic potential of cannabidiol from basic concepts to clinical studies. My name is Cherry Wainwright. I'm from the Robert Gordon University in Aberdeen in Scotland. I'm also the president-elect of the British Pharmacological Society, and I'm also a scientific advisor to Ananda Developments. And my co-chair is Professor Clive Page.
Good afternoon, everybody. My name is Clive Page. So I'm a professor of pharmacology at King's College in London. My interests are in inflammatory conditions, and like Cherry, I'm also I chair the advisory board for Ananda, and I'm also a non-executive director for Transparency. So, Cherry, over to you to make the introduction and the background to the meeting.
Thank you very much, Clive. First of all, a big welcome to both our audience and to our speakers today. And before we get moving, really, I'd just like to extend our thanks to Ananda Developments for their generous support, which has made it possible to present this webinar free of charge. And Ananda Developments is a life sciences company specializing in CBD-based therapies, and is actively advancing research with the University of Edinburgh and exploring new treatments for complex conditions. So if you'd like to know more about their team, then please visit the partnerships page on the event website for more details where you would be able to connect with them. I'd also really like to acknowledge the British Pharmacological Society for their support in organising this event, and we really do appreciate their efforts. Before we move into the science part, just to explain to the audience that there will be an individual Q&A session after most of the talks this afternoon. The Q&A will be conducted by using the Q&A box, which is at the bottom of your screen. So if you have a question that you would like to pose to the presenters, please put your questions in there, and we will endeavour to get those answered at the end of each talk. If we run out of time at the end of each talk, then we will come back to some of those in the panel discussion at the end. If you have any technical issues at all, please use the chat box because that is being monitored by the BPS team. And so if you have any problems, then they can respond and help you out straight away. So the reason behind running this webinar, well, cannabidiol is a non-psychoactive phytocannabinoid found in Cannabis sativa. And over recent years, it's received a lot of attention by scientists in trying to determine how it works, where it works, and what its potential therapeutic value may be. And as we'll come to hear through some of these talks, it actually has a very complex pharmacology. It acts at various targets and has significant potential in a broad range of disease states. The focus today is really going to be on the impact or the effects of cannabidiol on the inflammatory system. And so this webinar is designed to take us through from the basic understanding that we have around the molecular activity of CBD, but then to take us through to some very exciting clinical studies that are about to start, because as we build up the preclinical data on this fascinating compound, what we really need to do is to develop and move into some well-designed clinical trials. So I'm going to move on to our first speaker this afternoon, who is Dr. Katie Sloper, who is going to talk about cannabidiol as a multimodal pharmaceutical with broad therapeutic potential. Katie graduated from the University of New England, and then she received the University Medal at the Australian National University for her honours thesis, and then subsequently completed her PhD at that institution, where her focus was on cancer research. Following postdoc fellowships in both Australia and the United States, Katie has been working in healthcare across both public and private sectors, including as a senior policy advisor to the Queensland Minister for Health. And she's also a member of the Pharmacy Board of Australia. She's a medical researcher at Ananda Developments, and her work currently focuses on understanding the therapeutic profile of medicinal cannabis. So, Katie, when you're ready, please, if you would like to share your screen, we'll take it from there. Thank you.
Thank you, Professor Wainwright. I'll just share my screen. Did that share? Yes. Okay, well, thank you, Professor Wainwright. Thank you, Professor Clive, for the introduction. And thank you to the British Pharmacological Society for welcoming me to talk today. Welcoming me from sunny Queensland in the middle of the night. It's lovely to join you guys over in the UK. Now, I've been working with Ananda and MRX on cannabidiol, or CBD as I like to call it, on its potential as a therapeutic pharmaceutical for over 2 years now. And I'm still very excited and amazed at what comes up in the research. And today, I'd like to share with you, first up, a quick overview of CBD, highlighting its pharmaceutical potential. Then I'd like to focus on what we need to do to better exploit this potential to improve health outcomes. And then I want to focus on CBD's unusual versatility and diverse modulatory action and how it acts on multiple pathways and multiple targets. Just scrolling down. So, as Professor Wainwright mentioned, cannabidiol is derived from the cannabis sativa plant. It is, for medical purposes, obviously, though we need to use a pharmaceutical grade of CBD, but it is not the same as its well-known counterpart, THC. Unlike THC, quality CBD has a good safety profile and it's very well tolerated. And importantly, it's non-psychoactive and it's non-addictive, and it has minimal side effects. Yet despite this positive safety profile, and what you will see is its vast therapeutic potential, only one formulation has already been licensed, which is Epidiolex. And this one is used for seizures associated with Lennox-Gastaut and Dravet syndromes and tuberous sclerosis complex. So quite unusual. Now, for your information, if you're not aware, there are some cannabis-based medicinal product guidelines developed by the NICE Institute, but these ones were published in 2019 and updated in 2021. So they're probably due for a bit of an update soon, but I do refer you to those for more information on those guidelines. So the multitude, just on this slide, the multitude of pharmacological properties shown here in this slide illustrate CBD's potential. These properties stem from what CBD's diverse action on multiple targets and through them, multiple pathways. And I'll discuss that in more detail a bit later. But there is substantial preclinical evidence for most of these properties. And tonight, from other speakers, you will hear more about the anti-inflammatory properties, the anti-fibrotic properties, and the analgesic, as well as, and they'll be talked about a little bit later. But the point I want to make with this slide is that CBD possesses many more properties than are currently being exploited through its very limited use in very specific and very rare conditions. So what can we do to increase the route to expanding the therapeutic use of CBD? Well, obviously, the first thing we need to do is focus on more research, more preclinical studies and clinical studies. Preclinical studies, some of which we'll hear about today, includes mechanisms of action. Professor Oliver Wirtz will be talking about some anti-inflammatory actions of CBD, whereas Marta Baranowska and Anna Kraszewska will talk about its antifibrotic properties. In addition to that, though, we also need preclinical studies that also focus on dosage. To date, dosage in different research studies has varied considerably. At the same time, delivery methods and formulations have too. And formulations will be discussed in more detail tonight by Dr. Andrei Sitovs. In addition, we will also need well-designed clinical studies to fill the gaps in this knowledge and to convince the regulatory authorities of the safety and efficacy of CBD. At present, there aren't very many of those, and it's very difficult to convince the regulatory authorities of the value and the therapeutic benefits. However, tonight, Professor Marie Fallon will also be presenting on one of the current CBD trials already underway on chemotherapy-induced peripheral neuropathy. So with this next slide, I want to return to focus on CBD's modulatory action and how one molecule can exhibit so many properties. As you can see, there are a vast number of variable CBD target molecules. These are either largely membrane-bound or intracellular targets, and they can be both cytoplasmic and nuclear. As you can see too, they are far more extensive than just the endocannabinoid system. And it's through its actions on these molecules that CBD can exert its effects and influence and clinically assist in multiple diseases. So how can one molecule have so many therapeutic properties? As I mentioned with the previous slide, CBD acts on over 65 different molecules, but it also acts on multiple pathways. It also acts on different immune cells. And we and others believe, and there is evidence to support, that CBD is a modulator. It is not an on-off switch. So rather than completely inhibiting or activating any single factor, CBD may actually modulate a target or a pathway. I envision it like pistons within the cell. They're all being moved up and down at a different rate until a state closest to the homeostasis or to optimum state for the cell at that moment is achieved. In terms of its response, CBD is context dependent. By this, I mean that in a normal state, CBD may be latent, but when you put it under conditions of disease pathology, it actually can become active and may up or down regulate pathways according to the required response. And there is evidence, research evidence, that CBD responds differently in resting cells or healthy subjects to how it responds in stimulated or stress conditions. Now, my final point here is about contingency and redundancy. CBD is a bit like, works on like parallel or sequential circuits. It will target multiple parallel pathways, but at the same time, within a sequential pathway, it can target multiple molecules. And in so doing, it is actually allowing for contingency within its redundancy, so that if one target or pathway is defective, it can modulate the others to accommodate for that, to get to the nearest state closest to homeostasis. Now, in the next few slides, I'm going to illustrate this point in more detail. So this slide is very busy, I understand, but what I want to make is a couple of key points. If you look at membrane across here, you can see that CBD, which lies above it, can operate on each of these receptor targets. We have cannabinoid receptors, CB1 and 2. We have a G protein coupled receptor, GPR55. We have an adenosine receptor, a serotonin receptor, and a calcium channel receptor. And then down here, we have a nuclear transcription factor, PPAR gamma. And what you will notice is that all of these, which CBD works on, all the pathways lead to reducing inflammation here, reducing the pro-inflammatory mediators and increasing the anti-inflammatory mediators, and then subsequently having an effect on cell functions and mechanisms, decreasing neurodegeneration, proliferation, angiogenesis. But the point I want to make is that CBD is targeting multiple targets and multiple pathways to exert its effects, so that if one of these pathways happens to become defective, it is possible that CBD may modulate the others to try and retain some homeostasis within the cell system. Again, redundancy as a contingency plan. Another busy slide, but this one I want to point out something different. This slide shows multiple targets within how CBD operates on multiple targets within a pathway, multiple pathways that work to act on NF-kappa beta transcription of pro-inflammatory mediators. Here, I want you to focus on CBD, which is shown here in red. If you look at CBD, it's here in multiple places. On the left-hand side of the screen, it is working on the reactive oxidative species and inhibiting their production. It can also work on NF-kappa beta directly. And up on the right-hand side in the middle, it can work to prevent activation of NF-kappa beta. If we come over to the right side of the screen, CBD shown in red is working on STAT3, increasing its anti-inflammatory effects. And coming down to the left-hand side at the bottom, you can see CBD is also working on the production of pro-inflammatory mediators. What we are seeing is that it is working at multiple different spaces and type molecules within these pathways. So what we saw in the last slide, that it can work on multiple targets and multiple pathways and and have redundancy in that mechanism. It can also work on different molecules within the same pathway or multiple pathways and have redundancy in that. Again, if something goes wrong, it can work on another area to modulate the pathway to return to some sort of homeostasis. So my next slide also demonstrates that it has multi-layered approaches. So not only does it work on multiple targets, multiple pathways, it can also exert its therapeutic potential through multiple immune cells. If you look, CBD is shown in orange at the top of the screen, and it is working on most of immune cells, and it is working on them to influence the behavior of these immune cells and through them downstream signaling pathways and their effects on inflammation, apoptosis, and other cellular functions. So again, multi-layered modular approach, built-in redundancy as contingency planning. Now, my next slide, I've got a series of busy slides, but this slide is particularly important. It really highlights the modulatory functions of CBD and its context-dependent action. If you look on the left side of the screen, you can see that cannabidiol or CBD is shown here, and it can fluctuate here between antioxidant and pro-oxidant effects. Over on the right-hand side, we can see that CBD comes across, and it is modulating crosstalk between NRF2 and NF-kappa beta. And it is modulating crosstalk to modulate intracellular redox and inflammatory signaling. And in so doing, we can determine the cellular response under pathophysiological conditions. In this case, accompanied by oxidative stress. So CBD, through its modulatory behavior, switches between a number of effects to modulate depending on the context. So my final slide here is to show you that even through a small number of diverse receptors, CBD can have a significant influence on the body. Up the top is CBD. You have only a few of those 65 target molecules shown here. And there are varied molecules. You can see, as I said in that slide, they can be receptors, ion channels, protein coupled, and so forth. CBD acts on all of these to influence therapeutic or pharmacological properties. It acts on metabolism, anabolism, regeneration, inflammation, oxidative stress, neurodegeneration, and the list goes on. And through these therapeutic or pharmacological properties, it can influence different diseases. And again, we've got a vast number of those: immunological disorders, psychological diseases, neurodegenerative diseases, metabolic diseases, and other diseases. And a few examples of those are provided below. So what this really highlights in this slide is that CBD acts by modulating different targets, multiple pathways, multiple therapeutic effects, and then it can act on multiple different disease conditions. So it's quite a molecule. But what can we do to make sure that we can expand the use of CBD for it to exploit its therapeutic potential? As I mentioned before, we need well-designed clinical trials to determine the optimal dosing and formulation and administration. We need to know its efficacy for specific medical conditions. To underpin this, we need more research into its formulations with improved bioavailability. We look at its mechanisms in different diseases. That way we can have more precise and effective therapeutic applications. And finally, we also need to look at its interactions with other drugs. To conclude, I just want to say briefly thank you to the Emeritus Professor Cherry Wainwright and Professor Clive Page as the chairs, to my wonderful boss, Mr. Charles Morgan, who got me from Ananda Developments, who got me hooked on the non-addictive CBD, and to the British Pharmacological Society and Bonnie Rayner and her team for organising this event. I just want to finish with a slide from Shoutanek, who have very well encapsulated, I think, the need for what we do in the future with CBD. Thank you.
Thank you very much, Katie, for a really lovely introduction to such a complex molecule and all the potential that it has. I don't see if anybody has any questions. Please do put them in the Q&A. We don't have the facility for live voice questions, but I'll maybe start off with one. I mean, you've described CBD as this modulatory molecule that exerts this broad spectrum of effects. Do you think that CBD is quite unique when you compare it to, you know, any other types of molecule that is out there?
I do believe that there are other molecules within the, like endogenous to us, that do this. And I think that, for example, NRF2 is another example of a molecule that I believe has a modulatory type of function and capacity. But I think the uniqueness that comes with CBD is that we can add it to the body and it can modulate all those different factors. And yet it doesn't seem to harm, it just seems to adjust, whereas these other factors are already within the body. So what it provides us with is an external agent that we can add in as a therapeutic, and it can benefit people through its actions.
Maybe I can follow on to that, Katie. Thank you for a nice presentation. There are a couple of comments now that come through on the chat, and just an aside, it would be helpful if people have questions or comments, if you can let us know your affiliations so we can actually know who you are and where you come from. But you used a very nice analogy, Katie, about this behaving a bit like modulating pistons inside the cell, which I thought was very good. But given that this is a drug that clearly can interact with multiple targets, it is perhaps surprising that it has such a good safety profile from what we know of it so far, because many of those targets, when they've been targeted individually, have got all sorts of side effects and in some cases have limited their use. And I just wondered if you've got any comment on that.
Well, I think we have to be careful in looking at the research, particularly, is that even CBD has had some negative effects in some research contexts, but because it can be a modulatory function, have modulatory, I have difficulty with that word, have difficulty modulating other things. When you put it out of context, or if it may actually have a biological outcome different to the one we're measuring. So in some contexts, we may perceive that, say, for example, CBD might have a negative effect, whereas within the right context, it's actually having a positive outcome. Yeah. So, and I think that is because it is a modulator. So if you put it in an in vitro system and you're missing some of the things that modulates to adapt and get to a certain outcome, you may not see the outcome that you're actually measuring. For example, in an inflammatory setting, you might see a decrease in oxidation when you might want an increase in oxidation. And that's not a very good example, but if the body actually needs to say have apoptosis or something like that, you may actually require that outcome overall. Yeah.
Okay. So there are a couple of comments, Katie. So Andrew Brown, who described himself as ex-GSK and now a consultant, has asked that there's a company called Cannalife having an analogous CBD, which they're developing, which they described as 100-fold more potent. Do you know anything about that? And is it active at GPR55 receptors? Is that something you've know anything about?
I actually don't. But I would have to say that I would probably not be leaning towards something way more potent, because I think that part of the reason that what we're doing at the moment is the need for clinical trials is that you need to adjust the dosage. And
Yeah.
you also need to look at something that is also in tune with the levels that currently exist within the body. And I think that some of the studies have shown that if you go too high, you do actually get some negative effects occurring. And it is in some situations even biphasic CBD.
Okay.
So you have to be careful.
I think that question was actually quite relevant in terms of GPR55, because what we know, we've done a lot of work on GPR55 and which CBD is an antagonist. And we find that GPR55 is essentially a receptor that sits there and does very little under normal physiology. But as soon as you introduce a pathological state, such as, for example, metabolic dysregulation, GPR55 becomes much more active, and it produces its own downstream effects, but it also regulates the activity of other receptors through dimerization, for example. So I think that that's quite relevant, that question about GPR55. And that kind of, there's also Abraham, who's in Indonesia, has that might lead on to this, who's asked, we know that there are currently 2 cannabis-related receptors, CB1 and CB2. Do all the reactions you describe apply to both these receptors, or are there distinctive reaction differences between the two? That's a big question.
It is. So those 2 are the endocannabinoid receptors. And if you remember my slide, that there are over 65 target molecules that CBD will actually target. And in actual fact, its effect on CB1, the 2 receptors that are CB1 and CB2, is allosteric or slightly different. It has much stronger, CBD that we know has a much stronger impact on far more on other receptors, including GPR55. And so in actual fact, those receptors are not less potently activated, but they're not as predominant in the actions of CBD.
Okay, so perhaps related to this, Katie, there's another question from someone called Sal001. I'm not sure where they're from, but they're saying there's a lot of potential effects attributed to CBD. Do you expect this to be the same if CBD is given as an isolate or as part of a full or broad spectrum extract? And I suppose that begs the question about, you know, when you're giving the cannabis plant itself, you've got all sorts of other things in there, like terpenes, like other cannabinoids. There may be some interaction there that you may not necessarily see with CBD alone. So do you have any thoughts about that?
I do, and to be honest, I think that there probably are some interactions, and that when you have a phytocannabinoid, there may be differing effects depending on the terpenes or what combination you have it with. I do believe that when you put it with THC, CBD can actually counteract some of the negative effects of THC. And there are some drugs, as some of you would know, that are already out there that have THC and CBD combined, and they're already approved. However, I would say that based on my reading, if I were to take something, I would go with CBD alone. And I think that, having said that, I think there is some caution around synthetic CBD. I think you need to look at the phytocannabinoid itself and the context in which it grows.
Okay, so one final question till we keep to time. I mean, there are a few popping up now, and we'll let you have them, Katie, to answer perhaps afterwards. But Ben and Aidan from the University of Swansea have said some patents have now been filed for some of the CBD metabolites, such as 7-hydroxy-CBD for some fatty liver disorders. Is there evidence for different modulatory effects with the CBD metabolites compared to CBD itself? Do you think these metabolites potentially have any clinical use?
I haven't done a lot of reading on it, but yes, there has been some definite studies done on those metabolites, and they definitely have effects. Some of them will be more potent in some areas because you're obviously changing the chemical structure, and certainly I think some of CBD's actions stem from its hydroxyl groups. And so I think, yes, you may see some variations in their impact.
Okay, if you don't mind, I think we do have time just for one more. So this is from Helen Brooks, and she is asked, I noticed in one of the diagrams that CBD is able to treat psoriasis. Would this be via a dressing or cream? How effective would the treatment be? And is there any other potential skin conditions you think CBD could be used for?
Yes, there's actually some evidence that it is actually quite good for some eczema and some skin conditions and treatments of that. And in actual fact, it's funny, I just recently was talking to Mr. Charles Morgan, and we were looking at some photos of a dog that actually had some very severe skin issues, and they actually applied CBD oil on it, and it cleared up very, very quickly.
Okay.
So there's quite a lot of research evidence if you look for skin conditions and allergic and conditions, yes.
And I think you showed in one of your diagrams there are multiple inflammatory cells that are implicated in some of these skin conditions that actually CBD acts on. So, okay, well, I think, Katie, that's really good. You've had lots of questions, and I think it reflects you give a fantastic introductory lecture and an overview of what is really complex and interesting pharmacology around CBD. So thank you very much. You're allowed to go to sleep now, as it's the middle of the night for you. So thank you for your time. So we'll move swiftly on then, and it's my pleasure to introduce Oliver Wirtz. And Oliver is the Professor of Pharmaceutical Chemistry at Friedrich Schiller University in Jena in Germany. And Oliver's actually done a lot of work looking at the role that lipid mediators and other mediators play in the regulation of inflammatory cells. And Oliver's going to talk to us this afternoon about the anti-inflammatory effects of cannabidiol and some of the mechanisms behind that. So without further ado, Oliver, if you don't mind sharing your slides, and welcome, Oliver. We look forward to hearing from you.
Yeah, thank you very much for this very kind introduction. So good afternoon, ladies and gentlemen. I first want to thank the organizers of this webinar for setting up this important series of presentations about CBD, and for in particular giving me the chance to introduce you our recent discovery and report about this discovery of CBD being a modulator of lipoxygenases in order to promote the formation of inflammation resolving lipid mediators. So that's how CBD might act in order to resolve inflammation. So these lipid mediators play crucial roles in all stages of inflammation. So upon onset of inflammation by tissue injury, we have a rapid influx of neutrophils and monocytes that contribute to acute inflammation by producing leukotrienes and prostaglandins that are pro-inflammatory. They induce leukocyte chemotaxis, migration, cytokine production, degranulation, ROS formation. They increase vascular permeability, pain, and fever. And if these molecules persist for a longer time at high levels, they may eventually lead to chronic inflammation and organ dysfunction. So usually, however, acute inflammation is actively stopped and resolved. Tissue is regenerated. And here, in particular, the pro-resolving M2-like macrophages play a crucial role. And also this process of resolution and regeneration is regulated by lipid mediators, which are the so-called specialized pro-resolving mediators, SPMs, which are about 20 molecules encompassing lipoxins, resolvins, morations, and protectants. Two of them you can see here, and you can see that they are very similar in structure like leukotrienes and prostaglandins, but they have different receptors and opposite functions. They block neutrophil influx and activation, promote apherocytosis, phagocytosis, killing of bacteria. They sequester pro-inflammatory cytokines. They are organ protective. They promote wound healing and tissue regeneration. So it appears that this temporal lipid mediator class switch from pro-inflammatory leukotrienes and prostaglandins to these SPMs is crucial for stopping inflammation and for its resolution. So lipid mediators are synthesized from phospholipids, where first the phospholipase A2 is able to release the polyunsaturated fatty acids like arachidonic acid, EPA, or DHA, and they are then converted by lipoxygenases, cyclooxygenases, and SYP enzymes to monooxygenated products. And these are sort of intermediates, and based on the position where the oxygen is incorporated in the fatty acid, we differentiate between 5, 12, and 15 LOXs. And these monooxygenated precursors are in the second step again converted by lipoxygenases or epoxide hydrolases to make di or trihydroxylated products, and those are the bioactive lipid mediators. So in this context, arachidonic acid, as you have certainly seen many times before, is converted by cyclooxygenases towards prostaglandins or by the 5-lipoxygenase pathway with the helper protein flag to leukotrienes. Both molecules, as I mentioned, are pro-inflammatory, are produced by neutrophils, monocytes, and M1 macrophages. And interestingly, besides arachidonic acid, also omega-3 fatty acids like EPA and DHA can be converted by lipoxygenases, in particular by the 15-lipoxygenase. And together, these two enzymes, 5-LOX and 15-LOX, can make the so-called lipoxins. They can also make the E-resolvins and the D-resolvins. And the 15-LOX alone is able to make protectins as well as merasins. And merasins can be made also by the 12-LOX. Well, all these blue labeled molecules are pro-resolving and are termed specialized pro-resolving mediators, and they promote the resolution. So those are the good guys, and those are rather the bad guys, so to say. And the lipid mediator class switch means that during inflammation, prostaglandins and leukotrienes are downregulated and SPMs are increased in order to resolve the inflammatory process. Well, in order to study all those different lipid mediators, we choose macrophages because they can be easily produced by human monocytes from blood, differentiated and polarized towards pro-inflammatory M1 and anti-inflammatory M2 phenotype. And we tested what lipid mediators are produced by these cells, and we asked what would be an adequate stimulus in order to make lipid mediators. Well, just some basics. For a cell to produce lipid mediators, it requires, of course, the expression of the LOX and the phospholipase. It also requires a stimulus which is able to increase calcium and activate kinases, which then activate those two enzymes to move in the cell to an organelle, to a membrane compartment, where the PLA2 is able to release the fatty acid, which is converted then by the LOX to the lipid mediator. And actually, to make a long story short, what we found is then if we stimulate these macrophages with pathogenic bacteria, then the M1 make the pro-inflammatory prostaglandins and leukotrienes, but the M2 make these pro-resolving SPMs. And this can be easily seen without going into detail in this table. The M1 macrophage, after stimulation with E. coli, make mainly the 5-LOX and COX products, whereas the M2 make essentially these SPMs and 15-LOX products. And this is reasonable because the M1 phenotype expresses COX2, 5-LOX, and the helper protein, whereas the M2 phenotype clearly expresses the 15-LOX. And again, this is the key enzyme for formation of these SPMs here. So we could also show that these pathogenic bacteria are able to activate these LOXs. So you can see that the diffuse distribution of 15-LOX in blue in the cytosol changes towards an particulate staining. That means that the LOX moves to an organelle, and the same is true also for the 5-LOX that goes from inside the nucleus to the membrane. So how can we manipulate now this lipid mediator network in order to push resolution? One strategy, and it's the only available strategy actually, that attacks this pathway are NSAIDs, COX inhibitors, also glucocorticoids, that essentially block the COX enzymes. And that leads to a reduction of prostaglandins, but no beneficial elevation of those SPMs. In particular, PGE2 is affected. And as we all know, PGE2 is a striking pro-inflammatory molecule, but mainly in the acute phase of inflammation. It also mediates pain and fever. But we should not forget that PGE2 has also anti-inflammatory and pro-resolving properties, in particular in the late phase of inflammation, and also is gastroprotective and bronchodilatory. And blocking COX has side effects based on the reduction of these beneficial features. So an alternative strategy that we discovered is the modulation of these LOXes. And this is exactly what CBD is doing. But we discovered this strategy with another natural product. I want to briefly introduce it because CBD mimics these actions in a way that leukotrienes are clearly downregulated and these SPMs are increased. And this molecule is a natural product from frankincense, Boswellia serrata. And the product is acetyl-ketoboswellic acid, ACBA, and this was found to bind in an allosteric site of the 5-lipoxygenase. Here we have the active site, here the allosteric site, and we were wondering how could this compound bind here, but inhibit the activity of the enzyme in the active site. So this compound was long before described as a 5-LOX inhibitor, and in 2020, we identified the binding. And if you look at this area, we can see that binding of ACBA alters the interaction of those amino acids. It breaks it. So to make a long story short, ACBA breaks this interaction here between the amino acids and causes a domino effect towards the active site. And this changes the positioning of the substrate and therefore the oxygenation. With other words, ACBA reprograms the 5-LOX towards a 12-15-LOX. That means while 5-LOX usually incorporates oxygen at position 5, leading to leukotrienes and pro-inflammatory 5 heat, in the presence of ACBA, it changes the selectivity and incorporates oxygen at position 12 and 15, which are precursors for SPMs. And we tested that in neutrophils and M2-like macrophages from humans, treated them with ACBA, looked at the lipid mediator profiles, and found that, in fact, 15-LOX products are strikingly increased, in particular SPMs, and this is most evident in M2-like macrophages. So if we go a bit more in detail here, we can see that all the SPMs, protectants, merasins, resolvents, and lipoxins are strongly increased by ACBA and also the precursors produced by the 15-LOX. So this made us wonder whether this ACBA may also affect the 15-LOX, not only the 5-LOX. And therefore, we transfected hex cells that do not have LOXes with either 5-LOX, 12-LOX, 15-LOX1, and 15-LOX2, and incubated these cells with ACBA, looked at the lipid mediator profile, and to make a long story short, those hex cells that express the 15-LOX1, they are susceptible for ACBA to elevate strongly the formation of these SPM precursors. And in fact, we find that in a homology model, at least of the 15-LOX, ACBA binds to an allosteric site in analogy to the 5-LOX. So ACBA is not an approved drug on the market. It's a tool compound, but it's contained in commercially available frankincense extracts, and they can contain high amounts of ACBA and the related TBA. And we tested these extracts in M2 macrophages as well as in neutrophils. And to make a long story short, we see that also these extracts shift lipid mediator profiles towards these SPMs and 15-LOX products. So that means, in this context, ACBA reprograms 5-LOX to make a 15-LOX, but also directly activates 15-LOX. And we were wondering, how could we further enhance SPM formation? Well, this could be achieved by supplementing EPA and DHA as substrate, which is available as a product called Avalon that contains high amounts of lysine salts of EPA and DHA, omega-3 supplement. And when we combined these two, ACBA and EPA-DHA, then you can see for M2 macrophages, for instance, if you look at resolving D5, Avalon alone, no effect, boswellic acids extracts are active, and the combination is very active synergistically. And this is also the case in particular for RVE4. And we see these effects also in neutrophils, a very synergistic effect in terms of promoting SPM formation. And this combination we have patented, a combination of omega-3 and extracts of phosphatic species, and developed a product. And this product has been tested in a clinical trial in subjects with persistent knee pain. We treated these patients or subjects for 8 weeks with either placebo, frankincense extracts, omega-3 supplement, or the combination of both. And what we find is a very nice effect in terms of, for the WOMIC index, the arthritis index, a very strong improvement by the combination of the two versus placebo. The same is true for pain improvement, stiffness, and physical function. So why do I report about ACPA? Because actually we were wondering whether other prominent anti-inflammatory natural products that you can see here would have similar effects like ACPA. And we tested prominent representatives like EGCG from green tea, resveratrol, quercetin, patenolide, curcumin. And out of all those compounds, one popped up to be very interesting and effective, and this was cannabidiol. So in fact, cannabidiol sort of mimics the effects of ACPA, even though the structure is completely different. Cannabidiol reduces leukotriene formation, promotes the modulation or the activation of the 15 LOX in order to make these important SPMs like resolvins and protectants. And this publication here was highlighted by Derek Löwe very nicely in Science just after the publication. And as you know, CBD and THC as a mixture are available in Europe as an adjunctive treatment for spastic muscle paralysis associated with multiple sclerosis. And as you also know, pretty sure, CBD is simply the ring open version of THC, psychoactive, non-psychoactive. So we tested 8 different cannabinoids for their effects on these key enzymes in lipid mediator formation. And to make a long story short, you can see here the different structures. Here we have CBD, THC, for instance, and all of them actually inhibited the 5-lipoxygenase. Some of them inhibited the MPGS1, another interesting target, and most of them increased the activity of 12-15 LOX. This was a screening in neutrophils, and we then moved on to the M2 macrophages and incubated those with cannabinoids, stimulated these cells with Staph aureus, measured the lipid mediators, and what we can see here is clearly that CBD among those 8 different cannabinoids was the most active one to elevate the formation of the SPMs and the SPM precursors. At the same time, inhibited pro-inflammatory alpha-B4. So this means it pushes the lipid mediator class switch. THC is much less active, as you can see here. We then added cannabinoids simply to M2 macrophages without Staph aureus to see whether cannabinoids can actually induce this SPM formation in the macrophages, and it does. You see most of these cannabinoids were active to increase the SPM formation and precursors for about 300 to 400 fold. So here you can see the effect in many experiments that we collected on resolving D5, prominent SPM, which is increased by CBD, but not so LDB4. We then went back to HEC cells and tested whether cannabinoids could actually activate these different lipoxygenases in the transfected HEC cells. And as you can see here, again, those cells expressing 15-LOX1, they are very susceptible, having a 30-fold to 40-fold increase in the formation of these products by CBD. Also, in the structure of 15-LOX, we see that CBD, despite having a different structure from ACPA, binds in the similar site, in an allosteric site, and we could also show that this inductive effect of CBD on the SPM formation depends on the 15-LOX, as we use the 15-LOX inhibitor, BLX 3887, and you can see here for RBD5, CBD induces the formation of this mediator, and the 15-LOX inhibitor clearly suppresses it. So I pointed out that for the induction of lipid mediators, this translocation of lipogenases is very important, and we tested whether CBD could activate 15-LOX in the cellular context in terms of translocation. And what was very interesting is that within 15 minutes of CBD addition, we see that the 15-LOX from a cytosolic soluble distribution goes to membrane structures, similar as with pathogenic E. coli, but only the 15-LOX was moving, but not the 5-LOX, which is the pro-inflammatory lipoxygenase. And this was over the time not the case for CBD, but for pathogenic E. coli. And this LOX activation, 15-LOX activation, was independent of calcium. So we see no substantial release of calcium in these cells. And you can see that when we chelate calcium with BAPTA-ADTA, so no calcium, still the 15-LOX was moving when CBD was added, but not so with E. coli. And also still in the absence of calcium, as you can see here, resolving D5 is still induced by CBD. So then we also looked whether CBD induced SPM formation could be enhanced by supplementation of EPA and DHA, like we did with ACPA. And in fact, as you can see here for resolving D5, we have here the induction of CBD by CBD, no effect of avalum, but when we add both together, then we have a strong synergistic effect. And this also is the case for other 15-LOX products. So finally, we checked whether CBD could also be active in vivo in murine peritonitis to induce this SPM formation. And first, we used murine peritoneal macrophages to see whether murine cells, murine macrophages, are also susceptible to CBD. And you can clearly see that protectins, murasins are all increased, resolving D5 and the SPM precursors by CBD. We then moved on to the thymosin-induced peritonitis. We pre-treated the mice by intraperitoneal injection with CBD or indomethacin at 10 milligram per kilogram, indomethacin as a control. And we then induced peritonitis and collected the exudates after 4 hours. And after 4 hours, we see that cell infiltration was not blocked by CBD, but so by indomethacin. And we see, and this is very important, this lipid mediator class switch. That means that CBD slightly reduced 5-LOX and COX products, pro-inflammatory products, but increased the SPM formation. And this was, of course, not given by indomethacin. And here we have the single values. See CBD in green, increased protectins, murasins, resolvins, and the SPM precursors, while indomethacin rather reduces those molecules. If we look at the pro-inflammatory lipid mediators like 5-HEAT and LTB4, you see that CBD rather decreases those, whereas indomethacin elevates these. So to briefly sum up, CBD and other cannabinoids act as lipid mediator class switching agent that suppress leukotriene but promote SPM formation. CBD acts by allosteric 5-LOX inhibition and 15-LOX activation. We can combine CBD with EPA and DHA to get synergistically enhanced SPM formation. I think this could be very interesting as a therapeutic approach. CBD promotes SPM formation also in vivo in mice in thymosin-induced peritonitis. And as a take-home conclusion, so to say, we suggest that the suppression of these pro-inflammatory leukotrienes and the elevation of pro-resolving SPMs may underlie the anti-inflammatory properties of CBD. So I want to thank people in my group, in particular Paul Jordan and Lukas Peltner, who did most of the work related to CBD. I also want to highlight the contributions of our collaboration partners, Marsha Newcomer, Tonia Rossi, Charles Sirhan, and Giovanni Appendino, and of course also the funding agencies. And I want to thank the team again for setting up this webinar, and I want to thank you, kind people, for your attendance.
So thank you very much indeed, Oliver, for a very interesting presentation, and we'll open this up for questions if you don't mind. My first question to you, Oliver, as you know, there are also these very, very unstable hydroperoxyicosahexatetraenoic acids that do different things to the heats themselves. And I wonder because my experience of working with those, although they're unstable, they're actually pro-inflammatory, whereas the metabolites are actually, as you suggested, anti-inflammatory. And my second question or comment to you, Oliver, is that there's some very interesting work, as you know, from the group in Sherbrooke in Canada many years ago, that if you take leukocytes and platelets and put them together, you get these mixed 12-15 metabolites and 5-15. And I just wondered how you think that fits into that sort of hypothesis of a very clear distinction that you've given between the pro and the anti-lipoxygenase products.
Yeah, very good questions. So first of all, we have not covered these eats and diheats that you mentioned, which are in fact unstable. And in particular, there is an enzyme, the soluble epoxide hydrolase, that converts these epoxy fatty acids towards the diheats. And we tested whether CBD could interfere with this hydrolase, but it didn't.
Okay.
So we believe that these are not the molecules that are affected by CBD in this context.
Okay.
In fact, we have actually currently investigated whether CBD could also act on co-incubations of platelets and neutrophils, because as you mentioned, the combination, let's say there is a transcellular metabolism of fatty acids and intermediates between platelets and neutrophils, leading to very high levels of lipoxins in particular.
Mm-hmm.
So those cells possess enzymes that sort of synergize in producing these. And we are currently studying whether in these complex systems, CBD could also increase the formation of those anyway high levels of lipoxins. And our first data clearly indicate that this is the case.
Okay, well, that's very interesting, because as you know, platelet leukocyte aggregates are a very clear marker of systemic inflammation. If you could show actually that CBD through this mechanism reduces that number, I think that would be quite an interesting biomarker to look at in clinical trials.
Yeah.
So a completely unrelated question from one of the audience, Oliver, is from somebody called Roger Pinder. It's an interesting question. He says, is there any evidence that high exposure to frankincense, for example, amongst Catholic priests, is associated with a lesser incidence of inflammatory disease?
No, I don't think so. I mean, we have in the product that we developed, where we have frankincense extracts and this omega-3 supplement, we have quite high dosage of frankincense extracts, which is about 400 milligram, and you need to take 2 pills or 2 capsules twice per day.
Right.
So this gives like a gram amount of frankincense extract, and those extracts have very high contents of those phospholic acids. So you would not be able to have high enough levels just by handling frankincense in the church, so to say.
Right. Okay. So another question then from the audience. You, about halfway through your presentation, you showed us a table of the many different natural products that you'd screened as anti-inflammatory agents. And Dr. Gian Varden asked how you actually validated those screens, or what screens you looked at in order to come up with CBD. And related to that, I just wanted to add a question about anandamide, which as an endogenous endocannabinoid actually looks very similar to a lot of these lipoxygenase products. And I just wondered how that fitted into your sort of story.
Mm-hmm. So the screening system were the M2 macrophages. So we took the M2 macrophages and added those compounds at increasing concentrations and looked at the lipid mediator profiles, whether or not they are able to induce SPM inflammation. This was very simple. And among those, and I think that was a very important study to cover a broad range of well-known anti-inflammatory natural products like curcumin, resveratrol, and all the others that are known to have many, many targets. And it appears that these effects were mainly evident for boswellic acids and for CBD. There was another product which is called celastrol. This is from Trypteridium wilfordii. It's also a pentacyclic tritopane, very similar in structure like ACPA. So we believe that there are very strict structure-activity relationships. So that's the one thing. What was the second part of the question? Sorry.
It's about anandamide and whether
anandamide. Yeah. This is what we're currently doing. So we are studying a broad level of, let's say, cannabinoids or cannabinoid-like molecules for these activities on the 15 LOX. And what we find is that these actions of CBD are not related to the CB1 or CB2 receptor. So this is really evident. And for anandamide, we have ongoing experiments to investigate that, but it appears that they are not as active as CBD.
Okay. So my final question, I think there may be also a comment from Cherry, but one of the things showing your effects of CBD on the lipoxidase pathway, you've got a clear concentration dependent effect, but in some assay systems, because CBD can influence a lot of different receptor types, on some it's a partial agonist. So if you give enough of it, it will act as an antagonist. Do you ever see a U-shaped curve or a bell-shaped curve, or is it always just through a straightforward concentration-dependent effect?
Yeah, the latter is the case. So we see no U-shaped curve or banded curve. The point is that concentrations higher than 30 micromolar are cytotoxic to the cells, to the macrophages, and therefore we cannot go to higher concentrations. So the effective concentrations are in the range of 3 to 10 micromolar.
Okay.
So we have time for just one other question, and Marta Baranowska-Kutzo, who is actually one of our next presenters, asked the question whether CBD could be acting via GPR18 in resolving lipid mediators.
It might be possible. But I would be surprised. I mean, we have not tested that. And GPR18 is, to my knowledge, supposed to be a receptor also for SPMs, of some of the SPMs. But we have not tested that. It might be, but I do not see a direct correlation between the activation of the 15 LOX to make SPMs and binding of the SPM to the receptor.
Yeah, I'm not sure how much evidence there is that CBD activates that receptor anyway. I think there was a lot of confusion in the early days where what was described as a ligand for GPR55, 01602, is actually a potent ligand at GPR18, and I think that's added some confusion in about the two receptors and their involvement in certain things. So I think we're probably out of time for these questions. So Oliver, thank you very much for a very, very informative talk. And I do hope you'll be able to stay on to join in the panel discussion at the end.
Thank you very much.
We'll see you later. Thank you. So we move on now to our third presentation of the afternoon. It's a joint presentation by Dr. Marta Baranowska-Kutzo and Anna Krzeska from the Department of Experimental Physiology and Pathophysiology at the Medical University of Białystok in Poland. Unfortunately, they are not able to present here in person today for the very good reason that Anna is, I gather, undergoing her PhD viva as we speak. So she's been studying with Marta to perform her PhD on the evaluation of the effects of cannabidiol on inflammatory and fibrosis parameters in lung tissue and right ventricle in a rat model of monocrotaline-induced pulmonary hypertension. So they've kindly agreed to pre-record this talk. As a consequence, therefore, there won't be a live Q&A after this. But if you would like to post any questions, we will pass them on to Marta and Anna for them to consider. So I will hand over to the BPS team now, who are going to start the recording.
Ladies and gentlemen, good afternoon. First of all, I would like to thank British Pharmacological Society and Professor Terry Wright for opportunity to talk to you, and thank you that you dedicate your time to listen about the cardiovascular system and cannabidiol. My name is Marta, and I'm interested in cannabinoids and cardiovascular system since 2004, when I joined Professor Malinowska group and Professor Kozłowska. And Anna joined us a few years ago, and now she's finalizing her PhD thesis concerning cannabidiol and pulmonary hypertension. So, as I think you know, cannabidiol, CBD, is the second most prevalent from hundreds of active compounds derived from cannabis plants. It could be also produced in the laboratory, and Epidiolex is a pure synthetic CBD approved by FDA and European Medical Agency against the drug resistance epilepsy. Cannabidiol is also known because of its very wide therapeutical potency, including the cardiovascular system. And what is very important, according to report by World Health Organization, in humans, CBD exhibits no effects indicative of any abuse or dependence potential. And today, there is no evidence of public health related problems associated with the use of pure CBD. So why we are talking about the CBD in the cardiovascular system? The first reason is that the cardiovascular diseases are still the leading cause of deaths globally. 20.5 million people died from the cardiovascular diseases in 2023, reflecting 33% of all global deaths. Secondly, the cannabidiol has a very complex pharmacology also in the cardiovascular system. And thirdly, the determination of the action of CBD on the cardiovascular system is essential to be known, because there is still increased usage of this compound, both for therapeutical, also self-medication, and also recreational usage. There is also still increasing cover in the market and in the media, and you may see the cannabidiol just as a component of our morning coffee or post-workout smoothie, or even we can buy a CBD-infused grass. So that's why today we concentrate on the three aspects. I will tell you about the vasorelaxant and vasoprotective potency of CBD in essential hypertension, and Anya will introduce the antifibrotic potency in pulmonary hypertension. So to test the action of CBD on the local vascular tone, we use the isolated vessels. The human pulmonary artery obtained from patients undergoing the surgery because of the lung carcinoma, that's the margin of healthy tissue, and also animal tissues, the rat thoracic aorta, and the third branch of the main mesenteric artery, called G3 here, or a small mesenteric artery, reflecting the resistance artery. To reduce the probability of detecting changes related to given hypertension model, but not the hypertension itself, we use two models of hypertension, namely spontaneously hypertensive rats, or DOCA salt rats, mimics respectively primary and secondary hypertension in humans. The healthy control or normotensive control are Vistar Kyoto rats and sham rats. So in our hands, CBD relaxes human pulmonary artery concentration dependently. We also proved that almost all components of endocannabinoid system is present in the vasculature. Here I'm showing you the expression of CB1 receptors, CB2, vanilloid type 1 receptors, GPR18 or GPR55, located either in the endothelial layer or in the smooth muscle cell. So we performed a set of experiments to prove the mechanism, and what we found, the relaxation strongly depends on the endothelium and could be mediated via activation of prostacyclin receptor or prostanoid EP4 receptor, vanilloid receptor here, PPRS gamma, or calcium-dependent potassium channels. We didn't prove or didn't see the action of CB1, CB2 receptors or nitric oxide pathway. When we performed the post-hoc analysis of our patient characteristic, we found that in tissue isolated from patients suffering from hypertension, obesity, or hypercholesterolemia, we obtained the reduction of the CBD-mediated relaxation. So later on, we concentrate on the animal experiments on hypertensive tissue. Here, as you can see, in the vasculature of the hypertensive animals from this both model of hypertension I presented you before, we found the overexpression of on the gene level and on the protein level of CB1 and CB2 receptors. So the system is overactivated with overexpression of these receptors. We also found that in the two models of hypertension, we could see the relaxation to CBD. However, the efficacy of CBD-dependent relaxation strongly depends on the etiology of hypertension, and in this DOCA-SALT model, was stronger than in the control rats, and in spontaneously hypertensive rats was weaker compared to the control. It was corresponding also, the mechanism of this reaction corresponds to this overexpression of CB1 receptors, because as I marked here in the red boxes, in the hypertensive tissue, this relaxation depends on CB1 receptors, but not other types. Here I'm showing you CB2 or vanilloid type 1 receptors. So we already know that endocannabinoid system is overactivated in hypertension, that we have an overexpression of the CB1 receptors that may be responsible for vasorelaxation, and it could be the compensation reaction to the increased vascular tone in the hypertension. We also know that we have a variety of vascular complication in hypertension that could be both the cause or the consequence of the increased blood pressure. And the current treatment is aimed not only to reduce the blood pressure, but also to restore the endothelial function, restore endothelial attenuated vasorelaxation, enhance vasoconstriction in hypertension, or remodeling that happen in the hypertensive arteries. So in the next set of experiments, we concentrate if the cannabidiol given chronically could be vasoprotective. In the two models of hypertension, this SHR and DOCA-SALT, we were given CBD at the dose 10 milligrams per kilogram for 14 days, once a day. And then after that time, the animals were sacrificed and we collect the tissue. First of all, we looked if the CBD could affect the structural changes and endothelial dysfunction. Here, as you can see on the left side of the screen, in two types of vessels and two types of hypertension, we got a hypertrophy of media layer, and what is marked here with triangles, and as a red color, after the CBD treatment, we got the reversion of these vascular changes. And that was independently, as I say, of vessels, diameter, function, and etiology of hypertension. So we also looked at the expression of von Willebrand factor, which is a marker of endothelial dysfunction, and expression of endothelial nitric oxide synthase. Here I show you a photograph with arrows that make easier to follow the results. And as you can see, again, independently of the vessel size and the model of hypertension, we got the increase in arteries of expression of von Willebrand factor and decrease enos expression. And after the CBD treatment, we've got the reversion of the changes. So there was decrease on von Willebrand factor and increase of enos expression. It also correlates on the expression of enos on the gene and protein level. Secondly, we looked at the endothelium-mediated vasorelaxation and endothelial dysfunction, if the cannabidiol could help to relax vessels endothelium dependently. And here, as you can see, it improved acetylcholine-induced vasorelaxation. It happened independently of the vessel's diameter, function, and etiology of hypertension. We also looked if CBD could modify the vasoconstriction, and here what you can see that only in the vessels isolated from spontaneously hypertensive rats, but that was independent on the size and the function. So in aorta and G3, we got the reduction of phenylephrine mediated vasoconstriction. As I told you before, the CB1 receptors are involved in the vasorelaxation, and there is some kind of a feedback mechanism that this activation of this receptor through vasorelaxation could inhibit the vasoconstriction as a feedback mechanism. We also performed the experiments when we inhibit CB1 receptors by AM251, and what happened marked here again with these red boxes, we got the enhancement of the vasoconstriction mediated via phenylephrine. The results obtained here and this protective role of CB1 receptors corresponds also with the overexpression after the CBD treatment of the CB1 receptors in G3 arteries from spontaneously hypertensive rats mainly. Also, I would like to show you some unpublished results if CBD could modulate the expression of GPR18 or GPR55 receptors in this hypertensive condition in the vessels. And here again, you can see that independently on the diameter and function of the vessel and also the etiology of hypertension, we've got the overexpression of this both type of receptors here, and after the treatment with CBD, we've got the decrease of both types of expression. So to sum it up, the first part of the talk, cannabidiol in therapeutical doses given once and chronically has a vasoprotective effect regardless of the etiology of hypertension. And it happened independently of the diameter and function of the vessels in the context of reduction of vascular remodeling, improvement of endothelium-dependent vasorelaxation via increase in enos expression and reduction of von Willebrand factor expression. However, dependently on the hypertension etiology, it also reduces vasoconstrictive response to alpha-adrenergic 1 agonist. And cannabidiol has therapeutical potential as the adjuvant treatment of vascular diseases, in this case, essential hypertension. And now Anya will continue.
Good afternoon. My name is Anna Krzyżewska, and in the second part of the presentation, I will talk about anti-fibrotic potential of cannabidiol based on our own results in an experimental model of pulmonary hypertension in rats. Briefly, pulmonary hypertension is an abnormal increase in pressure in pulmonary artery above 20 millimeters of mercury in direct hemodynamic measurement. Pulmonary hypertension affects about 1% of the world's population, and current classification distinguishes 5 basic etiologic groups, which include pulmonary arterial hypertension, pulmonary hypertension associated with left heart disease, pulmonary hypertension associated with lung diseases and hypoxia, pulmonary hypertension associated with pulmonary artery obstructions, and pulmonary hypertension with unclear or multiple causes. In our research, we focused especially on pulmonary arterial hypertension because this is the type in which pulmonary artery lesions are a primary problem and therapeutic target. Pulmonary arterial hypertension is a complex condition with multiple underlying mechanisms. It includes an abnormal increase in cell growth within the walls of small pulmonary arteries. This is combined with endothelial cells dysfunctions. Specifically, endothelial cells start to produce more vasoconstrictors like endothelin or serotonin, while producing fewer vasodilators like nitric oxide or prostacyclin. In addition, muscular layer begins to thicken, and this process is often accompanied by the formation of blood clots, inflammation, oxidative stress, and structural changes known as plexiform lesions, which are a key feature of pulmonary arterial hypertension. Together, these changes increase in resistance in pulmonary circulation, putting more strain on a right ventricle. Over time, this process leads to right ventricle remodeling and fibrosis, which impairs its ability to function properly and ultimately results in heart failure. And as you can see here, 2 signaling pathways are involved in the molecular mechanism of RV fibrosis in pulmonary arterial hypertension, transforming growth factor beta pathway and WNT beta-catenin pathway. Trigger factors like inflammation, oxidative stress, or increased resistance in the pulmonary circulation activate TGF-beta pathway. TGF-beta binds with the receptors and activates and phosphorylates SMAD2 and 3. Phosphorylated forms of SMAD2 and 3 bind to SMAD4, and this complex migrates into the nucleus, binds with DNA, and regulates the transcription of target genes like collagen 1 or fibronectin. WNT beta-catenin pathway is also activated by previously mentioned trigger factors and TGF-beta. Normally, in healthy condition, in the absence of WNT ligands, beta-catenin is phosphorylated by degradation complex. When WNT ligands bind to receptors, beta-catenin, the ability of degradation complex to phosphorylate beta-catenin is inhibited, and unphosphorylated form of beta-catenin migrates into the nucleus and indirectly regulates the transcription of target genes like cyclin D1 or c-Myc, which is important for cellular proliferation, differentiation, and survival as well. Currently, we use 3 groups of drugs in the specific treatment of pulmonary arterial hypertension, drugs affecting nitric oxide pathway, like stimulators of guanyl cyclase and inhibitors of phosphodiesterase type 5. Drugs affecting endothelin pathway, like antagonists of endothelin receptors, and drugs affecting prostacyclin pathway, like prostacyclin receptors agonists. These therapies improves patients' quality of life, but do not fully cure the disease, and their mechanism of action is often limited to the vasodilation effects, vasodilatory effects. And I would also like to mention here about novel therapy targeting activin and TGF-beta signaling pathway. So sotatercept is first in class activin signaling inhibitor, which modify damaged pulmonary arteries by inhibiting their growth. And considering the fact that cannabidiol has so many beneficial properties as presented previously by my colleague Marta, and the fact that there is a lack of effective treatment of pulmonary arterial hypertension, the aim of the studies was to evaluate the effect of chronic administration of cannabidiol in a preventive model of monocrotaline-induced pulmonary hypertension in rats. And I will only present the results on the effect of cannabidiol on cardiac fibrosis in pulmonary hypertension model. The experiments were performed on previously collected and properly secured tissues. To obtain pulmonary hypertension, rats monocrotaline was injected once at day 0. Then healthy and pulmonary hypertension rats were given cannabidiol at a dose of 10 mg per kilogram of body weight intraperitoneally for 21 days. In this way, we have obtained 4 groups of animals: control treated with CBD, monocrotaline, and monocrotaline treated with CBD. The fibrotic parameters were evaluated using ELISA, Western blot, and immunohistochemistry methods. And as you can see here, in monocrotaline-induced pulmonary hypertension model in rats, we observed increased right ventricular systolic pressure, increased Fulton index, increased lungs weight to body weight ratio, decreased oxygen saturation, and increased wall thickness of pulmonary artery. And chronic CBD administration decreased RVSP and wall thickness, did not affect Fulton index and lungs weight to body weight ratio, and improved oxygen saturation. As you probably know, NT-proBNP is considered to be one of the most important indicators useful in RV diagnosis in pulmonary hypertension patients. And as you can see here, NT-proBNP plasma level was elevated in the monocrotaline group, and chronic CBD administration decreased this plasma level. In monocrotaline group, we also observed increased cardiomyocytes width and interstitial and perivascular areas of fibrosis. Areas of fibrosis are marked by a blue color here. And chronic CBD administration decreased cardiomyocytes width and interstitial and perivascular areas of fibrosis. Fibroblasts are the main cells that produce collagen after RV injury, for example, pressure overload. And as you can see here, in monocrotaline group, the immune expression of fibronectin and fibroblasts was elevated. Immunoexpression of fibronectin and fibroblasts is marked by a dark brown color. And after CBD administration, the immunoexpression was decreased. In monocrotaline group, we also observed increased TGF-beta1, galectin-3, SMAD2, and alpha-SMA parameters. In contrast, VE cadherin in this group was decreased. VE cadherin is a marker of healthy endothelial cells. And chronic CBD administration decreased TGF-beta1, galectin-3, SMAD2, did not affect alpha-SMA, and increased VE cadherin expression. And here we have a brief summary. We suggest that beneficial anti-fibrotic potential of cannabidiol may be due to the inhibition of TGF-beta dependent pathway. TGF-beta activates fibroblasts and activates their proliferation. Activated fibroblasts transform into myofibroblasts, which express alpha-SMA. And mature myofibroblasts start to produce collagen and fibronectin, which can lead to RV fibrosis. At the Pulmonary Hypertension World Symposium this year, we presented our preliminary results suggesting CBD's involvement in the inhibiting beta-catenin signaling pathway in monocrotaline-induced pulmonary hypertension in rats. As you can see here, we did not observe the differences in expression of beta-catenin between groups, but as you can see here, beta-catenin marked by a dark brown color is located especially in the intercalated discs, and in monocrotaline group, the expression of beta-catenin is located also in the cytosol and nucleus. And after CBD administration, this image is quite similar like in control group. As I mentioned earlier, beta-catenin translocates into the nucleus and activate target genes like cyclin D1. And as you can see here, we observed increased expression of cyclin D1 in monocrotaline group, and after CBD administration, the expression was decreased. To summarize our and the other authors' results, in an experimental models of pulmonary hypertension were observed these changes, and chronic CBD administration at a dose of 10 mg per kilogram of body weight decreased right ventricular systolic blood pressure, decreased remodeling of pulmonary arteries, lungs, and RV, decreased oxidative stress and inflammation in lungs and arteries, inhibited RV and arteries hypertrophy, improved oxygen saturation, exhibited protective role on endothelial cells, and normalized plasma coagulation and fibrinolysis parameters. As I mentioned, sotatercept is an inhibitor of activin, and in this way inhibits the proliferation of small pulmonary arteries. And it is worth mentioning here that cannabidiol, in addition to its vasodilatory, antioxidant, and anti-inflammatory properties, appears to target the similar signaling pathway as sotatercept, I mean SMAD2. And in conclusion, cannabidiol has potential to inhibit right ventricular remodeling, possibly by regulating transforming growth factor beta and beta-catenin signaling pathways. And cannabidiol has therapeutic potential as the adjuvant treatment of vascular diseases, in this case, pulmonary arterial hypertension. And due to the fact that our presentation is recorded, please feel free to contact with us if you have any questions about the first or the second part of the presentation. Here you can find our email addresses, and we also would like to thank our colleagues and our team from the Department of the Experimental Physiology and Pathophysiology, the Medical University of Białystok in Poland. Thank you for your attention.
Thank you very much.
So thank you very much, Anna and Marta, for a really interesting presentation. And
Fabulous, yeah.
because it was pre-recorded, as they said in the last slide, please do send any questions in and we can make sure they're passed on to the authors. And we wish Anna well with her thesis defense. I hope by the end of this meeting, she'll know whether she's got her PhD or not. So in order to keep to time, we'll move on to the next presentation. And it's my pleasure to introduce Professor Mary Fallon from the University of Edinburgh. Mary's the Professor of Palliative Medicine in the University of Edinburgh, well known internationally for the work that she's done in this area, particularly around pain control. And Mary has also led a number of randomized controlled trials looking at new interventions for the treatment of pain, particularly in what she's going to talk about today, chronic chemotherapy-induced peripheral neuropathy. And one of the things about Mary that many people won't necessarily know is that she also spends a lot of her time helping lower middle income countries actually access appropriate analgesia for different types of pain. So Mary's going to talk to us today about the ACTION trial that she's setting up and running in the University of Edinburgh to investigate CBD as a potential treatment for this particularly nasty form of chronic pain. So Mary, over to you.
Thank you very much, Clive, for the invitation, and thank you also to Cherry as well. So in the next 20 minutes or so, I want to explain how we've come to the point of actually attempting a clinical study using CBD. And this is about the management of something called chronic chemotherapy-induced peripheral neuropathy. And the study is called the ACTION trial. Right. So first of all, just very quickly, a little bit about chemotherapy-induced peripheral neuropathy. It's a potential side effect for any patient receiving neurotoxic chemotherapy. And while it's got sensory, motor, and autonomic components, there's definitely strong evidence that the dominant factor is the sensory components. These tend to be acute during receiving chemotherapy and the days afterwards. And then they can be chronic, and chronic is defined as any patient with persistent neuropathy 3 months or more after the last chemotherapy. And patients will often describe these sensations as very strange and distressing. It's very much in keeping with standard neuropathic pain. One has to delve into it to find out the descriptors. The other very relevant issue here is patients will sometimes develop something called brain fog. And this is particularly troublesome for younger patients who are maybe more sensitive to the onset of this, and they just feel slightly dimmed, slightly obtunded a lot of the time, and just not functioning the way that they would usually function. So our systematic review meta-analysis showed that up to 50% of patients who've received neurotoxic chemotherapy will actually have ongoing chronic SIPN 12 months after treatment, can be very debilitating and have a major impact on quality of life. The other important thing is it can limit future cancer treatment. Someone who's living with very difficult peripheral neuropathy who may need further treatment may have difficulty receiving that treatment because of the potential of just not tolerating it because of worsening the neuropathy. I wanted to put this up for 2 reasons. And the first thing is, obviously, with a peripheral pain, and the spinal changes that talk to the brain, that tells you where it is and how severe it is and what it's like. The brain also talks back to the spinal cord, and you have the circuit of interdependence and exacerbation, and mood, sleeplessness, etc. will very much feed into the exacerbation of these problems. The other thing, as I've mentioned, is where there are situations of so-called brain fog due to the central neuroinflammation caused by chemotherapy. This obviously can exacerbate the whole situation for an individual patient. The management is very challenging. There's low-level evidence for duloxetine and SNRI. There's reasonably good evidence for physiotherapy for peripheral small fiber exercises. Drugs such as opioids are not helpful, and often they would be not appropriate, even if we did feel they might be potentially helpful. These are patients who are going on to live, hopefully, cancer-free after the treatment with chemotherapy, and chronic opioid use we would try and avoid at all costs. The mechanism for peripheral neuropathy has very strong evidence for a neuroinflammatory component, as well as strong emerging evidence for oxidative stress. So I just want to concentrate in the middle point. One of the challenges of looking at chronic neuropathic pain is it's often in the clinical laboratory, if you like, you're looking at patients who actually have quite complex problems by the time they specialist clinics and by the time they enter into clinical studies. And one of the very positive things about chemo-induced peripheral neuropathy is using patients who've received what we call adjuvant treatment for cancer. So that's this preventative treatment. It's often referred to in lay terms. These patients, after their adjuvant treatment, will in fact go on to be cancer-free and well. And they're not on this sort of analgesics that other patients, particularly with active cancer, will be on, such as opioids and what we call adjuvant analgesics, such as amitriptyline, pregabalin, etc. So peripheral neuropathy is a model to study where you can get rid of a lot of noise, is very good. And from the point of view of a model that has the basic science neurobiology that fits the potential mechanisms of action of cannabis-based medicine, it's a very, very good fit. This, in fact, comes from Professor Clive Page's laboratory. We've heard some excellent talks today. I think Katie summed some of the issues up in our patient group very nicely. And that's that we're really looking at one target. Our patients generally, whether they have active cancer or whether they're in the cured cancer group with ongoing pain, it's a soup of neurotransmitters that we're actually dealing with. It's a soup of neuroinflammators, etc. And this summarizes some of the evidence that's all totally relevant to peripheral neuropathy from the preclinical point of view. So the proposed study is a phase 2 randomized controlled trial of oral cannabinoid, CBD, versus placebo in the treatment of chemotherapy-induced peripheral neuropathic pain, hence the acronym ACTION. Now, the first thing I want to say is we chose a crossover design, and we chose a crossover design because it's well documented that placebo design in neuropathic pain tend to result in negative studies. You get a very, very, very high placebo response with the parallel design. And the other thing that we've done, as well as the crossover design, where if you like, each patient is their own control, We're also giving a drug, which will be placebo. We will know that, but the patient won't during the 2-week washout. Neither the team nor the patients will know what they're getting in the 2 crossover phases, but during the washout period, we are going to continue with the placebo, which again is to try and talk to this issue of minimizing placebo response. So the rationale for, what we want to do and why we want to do it. NICE in the UK have done reviews for the evidence of cannabis-based medicines, and they came up with the same conclusions as the International Association for the Study of Pain, IASP. And the bottom line was they both saw that there was very strong preclinical evidence, particularly that matched some pain syndromes, but that the clinical studies conducted until this point were poorly conceived, poorly designed, and really were set up to fail from the beginning. So they both said there is a lack of evidence rather than no evidence. And IASP in particular have given very helpful recommendations for the methodology that should be used in cannabis-based medicine trials. NICE have put out a call where they're particularly interested in CBD alone, or if necessary, very small amounts of THC, but the preference is CBD alone. We're also in the work using the optimum guidelines for assessments of peripheral neuropathy trials, and we've added in mechanistic assessments. I'll come on to that. Pilot work that we've done over several years in Edinburgh has helped us to understand the clinical group of chemo-induced peripheral neuropathy, and also we've had some experience with using CBD in individual patients. Of course, last I've got here, understanding why neuropathic RCTs fail, not just in cancer patients, but in any sort of pain study. And we've tried very much to incorporate that and improve on that in our study design. So the specific recommendations from IASP for cannabis-based medicine studies are you've got to match the pain you choose to study with the preclinical evidence of neurobiology of pain, mechanisms of analgesia, and the mechanisms, the believed mechanisms of action of cannabis-based medicines. The other thing that we have to be very careful about is looking at the outcome measures, and these must capture the clinical picture. So unlike in the laboratory, where we're maybe looking at one endpoint, in patients, one endpoint, actually, the unity of one endpoint will often translate into a bundle of effects for a variety of reasons. Pain might have a knock-on effect on sleep. It may have a knock-on effect on anxiety and mood, appetite, and of course, physical activity. The potential mechanisms we've embedded within the actual study. So our research question has been, what is the efficacy of a broad spectrum CBD oral tincture containing CBD isolate and terpenes in patients with neuropathic pain due to SIPN, and what are the potential mechanisms of action? And understanding the mechanisms very much helps us to understand the patient-reported outcomes, and also clearly to help us move forward with phase 3 studies. So this phase 2 clinical trial, we're very grateful to Ananda, who are providing the IMP, which is MRX1. And the study will be conducted just within Lothian, within one health board at this point, at the phase 2 point. And it's been funded, fortunately, by NIHR, EME, with over £1 million grant. And we believe that recruitment into the study will be easily achievable because we work very closely with our oncology colleagues. We already have clinics for patients with chemotherapy-induced peripheral neuropathy, and sadly, it's a very common problem with no effective treatment. So, going on to understanding the treatments, I was fascinated to hear Katie's talk and looking at the different targets and pathways and mechanisms. I think one of our overwhelming problems when treating neuropathic pain, and particularly this chemotherapy-induced neuropathic pain with complex pathways and drawing in this real soup of neurotransmitters, is that we often treat patients with single management approaches. So, for example, we use capsaicin TRPV1. We do use that. We use both the cream, but we also use the high-dose patch. And it will work in some patients and not in others. Clearly, the 5-HT family of drugs, we use these too. And the overwhelming problem, we believe, is that we have a number of complex pathways activated by this neurotoxicity in the peripheral nerves, and no one target is actually going to solve the problem for your average patient. So that's the positivity within cannabis, CBD in this instance. These are just going through the formal aspects of the study. The principal aim is to determine the efficacy of CBD, isolate and terpenes in patients with SIPN. The clinical objective, following IASP guidelines, we very much stuck to how do we best measure peripheral neuropathy, the sensory component, the painful component, and that's using the EORTC SIPN questionnaire that was specifically designed for this particular problem. And then the primary mechanistic objective, because we're going to have embedded fMRI, we'll also have, CBD levels, endocannabinoid levels, cytokines, quantitative sensory testing. But from the fMRI point of view, we'll look very much at the anterior cingulate cortex connectivity in those receiving active treatment during resting state fMRI. And we'll look at any associations with changes in clinical pain and well-being scores. And then, of course, there's the very light touch look at the health economics. This is the design here. So the target recruitment is 46 patients per group, and they will cross over after 7 weeks. So that's 5 weeks of active treatment or 5 weeks of placebo, the 2-week washout, and then they'll switch to the alternative, which will be either 5 weeks of active or 5 weeks of placebo. During that first phase, patients will have an fMRI scan at baseline and at 5 weeks, and they'll have blood analysis at regular time points. We very much are trying to get meaningful patient-reported outcomes, and we'll be looking at sleep, we'll be looking at mood, anxiety, and of course, physical activity. Overall quality of life is designed for the EORTC SIPN tool. And of course, patient perceived effects, side effects, and any serious adverse events. These are just the primary and secondary outcomes in more detail here. And I won't go over that again. So I just wanted to mention, the whole problem about clinical pain studies again, because it's relatively easy. Well, it's not easy. It's always challenging to do clinical research, but it's easy to do the wrong research, and it's easy to be excited about it and think you've got it right. But to actually get it right, particularly in neuropathic pain studies where we know there's a high placebo response, it's so important to match the pain, in this case the neuropathic pain, with the preclinical evidence of the neurobiology of that pain and the mechanism of the proposed analgesia. And we think here we've got a good match. We've got a neuropathic pain that's driven through neuroinflammation and oxidative stress. We've got a CBD where we believe one of the main mechanisms of action is going to be anti-neuroinflammatory, antioxidative. We believe that we're quite sensible about the outcome measures in that we're looking at both patient-reported outcomes and we're looking at mechanistic outcomes. But in the patient-reported outcomes, we're trying very hard to capture that bundle of effects rather than a unit of outcome, just improvement in pain per se. We do believe that the use of the fMRI will be really interesting in disentangling some of the placebo and analgesic and other drug responses. One of the things we've learned from other pain studies is that obviously pain is by definition a shifting paradigm. As soon as you improve a patient's pain, they're likely to do more, and when they do more, that can bring the pain back to the starting point. But they might be quite happy with that because their quality of life is improving. And that's something that we very much cater for within this design, looking at activity and overall quality of life as well. We see problems with, well-known drugs such as pregabalin in our patient group, where often patients get a great anxiolytic effect initially. They sleep better, and you think pain is improving at a relatively small dose, and of course you quickly lose that. And so understanding the various mechanisms of the CBD in relation to our pain paradigm is really important. And of course, from the cohort literature, we know that cannabis-based medicines lead to a mixture of improvements, this sort of bundle that I've referred to. You often, yes, do get a bit of improvement in pain, but improvement in sleep, improvement in anxiety, and it often leads to a marked increase in general quality of life. So this combination of outcomes, we hope to capture very, very carefully in our statistical analysis plan. It will be a predetermined outcome. It won't be ad hoc, and we hopefully can speak to that in quite a robust scientific fashion. I'd like to thank very much my co-applicants in the NIHR EME application, and particularly Heather Wally, who's joint lead applicant and a neuroscientist in Edinburgh, and of course our PPI co-applicant, Constantine. It goes without saying a huge thanks to Ananda for their patience while we got this funding and for agreeing to supply MRX. Thank you very much.
Thanks, Murray, for a superb talk.
Yeah,
Excellent.
thank you, Murray. So Murray, obviously this is a really important trial because I think we heard from Katie at the beginning, there's a need clinically for good placebo-controlled trials. And clearly there's a lot of effort gone into thinking about how to have the right outcomes, what to measure. And whilst this is a pilot study, I think you've highlighted some of the issues. But actually there was a question put up earlier to Katie that we didn't get around to, but I think I'm going to put it to you, Murray, because I think you'll have some views on this. This is from Dr. Shishka Gallo, who said, really, should we be looking at CBD on its own when, as you've said, many of these patients are potentially taking cannabis itself, which is a lot more than CBD. And there are a lot of different things that can actually influence that, like stress, diet, body weight, etc. And there is the entourage effect between the various things in the plant. And do you think, therefore, that we might lose some of that activity when you move from cannabis to CBD itself, albeit that there may be terpenes there? And I suppose related to that, a bugbear of mine, Mary, which I may have shared with you previously, but I think the cannabis-based medicine to most people, and particularly physicians, equals THC, which, as we've heard, well known is psychoactive, and very often I don't think is necessary for some of the pharmacological actions we've heard about. And should we be getting away from this term now? Dr. Gallo has suggested that is it fair to put CBD into pharmaceutical research at all because it's part of a wider set of molecules in cannabis? But I just wondered what your thoughts were, because you mentioned you've had experience of patients who've been taking both the cannabis plant but also CBD.
Yeah. So thank you, Clive. There's lots of things there. So I don't particularly like the term cannabis-based medicines, and that very much has come from IASP because they didn't define that we should be looking at just CBD, although interestingly, NICE are much more interested in CBD alone and not THC. And to deal with that, I would say two things. The first thing I'd say is the preclinical evidence in CBD is actually very encouraging. And that, married together with the societal aspects, is actually really important, because we've been through all the issues with opioids and the bad press there. And there's no doubt that with THC, you're more likely to get side effects. I've done work in the past with Sativex and cancer pain, and some patients do get side effects with THC. Similarly, those patients that go out and source THC, CBD, cannabis themselves, again, are more likely to get side effects. And I'll come back to that in a moment. And I think the clean way forward for both the science, the clinical research, and the benefit of society is to stick with CBD plus whatever terpenes in the first instance. And, you know, if we're disappointed, we're disappointed. I don't think so. I think that's the way forward. Patients who source what's available, either, you know, buying it on the internet or, you know, sourcing just cannabis, the outcomes are often disappointing. And they might feel that first of all, those that buy on the internet, I don't think what's available is predictable and consistent enough. And that's been a bit of a problem because our patients are spending a lot of money in this. By the way, I should say, we will screen for anyone using, you know, cannabis at baseline and through the study so that we know what's what. So I think it's disappointing when patients buy it, and when they source and smoke, you know, marijuana or whatever, again, they're more likely to get side effects. I've recently managed 2 patients with very profound, well-established, very difficult chemo-induced peripheral neuropathy, off-the-scale pain with CBD alone, and in therapeutic doses, and they both had dramatically wonderful outcomes at the first starting dose. These were patients that I wouldn't enter into clinical trial because I would regard them as not within that normal spectrum and very, very, very profound problems. And both of these ladies had had breast cancer and been treated successfully with chemotherapy, but the taxanes lead dreadful neuropathy. And both had tried internet-sourced cannabis with no effect, but had profound benefit with CBD. So my feeling is we should stick with the plan. And certainly as far as my clinical preference goes, I prefer to have CBD without THC. and optimize the dose with minimal side effects, and work with that within a clinical trial, rather than being tempted to have a small amount of THC.
No, that's very clear, Marion. I thought I'd know your answer, but I think that's really, really clear. I mean, but related to that, there's a question come up from Abraham in Indonesia, actually going back to saying therapy with medical cannabis is usually personalized. What about clinical trials? Are there out there to support that? And do you have to make dose adjustments for each patient? I think you've already made this point away, Mary, that if you're buying stuff on the internet or even illegally as cannabis, you have no idea from batch to batch whether it's characterized and validated and consistent. So, but is there anything further you'd like to add to Abraham?
Yeah, I mean, I should have said that within the clinical trial we're doing, there will be a dose titration. Though it won't be forced, which means that you have your agreed starting dose, and depending on pain outcome, so if the patient has a marvelous response to the first dose, they would just stay at that dose. But if they don't have a response and they don't have side effects, they would go up to the next dose range. So we would have a weekly increase in dose, and that hopefully will give us an idea in relation to this question of the dose ranges that are likely to be useful in clinical practice, and also if there's a particular dose at which patients get side effects. But I think his point that it will ultimately be individual. It's like all drugs that we use for pain. They're very individual. We have a maximum dose that we would titrate to usually, but the dose you use is always very individual. And more often than not, it's the side effects that limit dose titration. And that's another reason that it's good not to have the THC in it, because it'd be a shame if, you know, analgesic effect through limitation of dose titration was attenuated. Obviously, we will have some blood samples, and we'll be able to look at levels at the given dose range for that a patient ends up on, and that too will be interesting. You know, it's phase 2, it's only 90 odd patients, but I think it's enough to start giving us a flavour.
Well, I think it's a properly controlled study, and that's the important thing. We wish you well, and we hope for every success with the trial. So thank you again, Marion, and we'll move on then to Cherry's going to introduce the last speaker, which I think is another important issue, is the challenges of giving oral cannabidiol therapy and the selection of doses and dose range. So over to you, Cherry.
Well, you actually took the words right out of my mouth there, Clive. I was going to say this segues very nicely into our final talk of the afternoon, and that is, you know, as you can create as much preclinical data as you wish, but the first question a pharmaceutical company will ask about a potential novel drug is it orally active? And cannabidiol, as we are about to hear, has quite a complicated story around its oral bioavailability. So I'm delighted to introduce Dr. Andrei Sitvos from Riga Stradins University in Latvia. He's a certified pharmacist, but also a researcher in with particular interests in bioavailability, ex vivo permeability studies, pharmacokinetics, solid dosage forms, etc. And he's done a very thorough analysis of the challenges that are faced around oral cannabidiol delivery. So it gives me great pleasure to introduce Andrei, if you would like to start sharing your screen.
Thank you for this introduction, dear audience, dear British Pharmacological Society. Thank you for the invitation. It is a pleasure to share our findings with you. As a formulation scientist, I represent the leading research group of the pharmacy of the Faculty of Pharmacy at Riga Stradins University in Riga, Latvia. and we work extensively with formulations to improve the solubility of poorly soluble drugs. And cannabidiol happened to be one of the poorly water-soluble drugs. So we were interested in reviewing its properties in the context of peripheral dosage forms. As we have already heard today, this non-psychoactive cannabinoid obtained from cannabis plant is being used as a prescription drug and as a food supplement, but of course the oral dosage forms, oral administration prevail. Major challenges that CBD oral formulations are facing originate from its chemical instability and low oral bioavailability. Objectives of our literature study were to investigate the cannabidiol physicochemical properties that define the oral bioavailability, such as the pKa, log P, or partition octanol water partition coefficient, solubility in different solvents, melting points. Then we looked at the CBD from the point of view of biopharmaceutics or developability classification system, considering its permeability, solubility, and those aspects. Then we evaluated cannabidiol permeation and absorption from the gastrointestinal tract into the systemic circulation, and we checked some most important pharmacokinetic parameters of cannabidiol. Cannabidiol belongs to prenol lipids, aromatic monoterpenoids, and it's a highly lipophilic compound. It is easily degrading under the UV radiation and different oxidants in acidic and alkaline environment. Upon storage or at higher temperatures, 50 to 80 degrees Celsius, cannabidiol substance degrades approximately 1% a day. The optimal pH for CBD stability is around 4 to 6, but in very acidic environments, cannabidiol undergoes cyclization, reaction, and forming the well-known psychoactive substance delta-9 tetrahydrocannabidiol. And this reaction was also reported to take place in acidic environment in the stomach in vivo. Regarding the physicochemical properties of cannabidiol, its melting point is approximately 66 degrees Celsius. As I have already said, this is a highly lipophilic substance, so log P is around 6. The pKa value is around 9, which classifies cannabidiol as extremely weak acid. It is practically water insoluble, but it's very well soluble in different organic solvents and different oils, and the highest solubility was reported in medium chain triglycerides and also in different eutectic systems. Eutectic system is a mixture of different solvents where the melting temperature of the mixture is lower than the melting temperature of each individual component of the system. From the point of view of classification of lipophilic substances that could be grease balls or brick dust, cannabidiol substance can be classified as grease balls. It has high lipophilicity, but at the same time relatively low melting point. That means that if we want to improve the solubility and permeability of this drug, different lipid-based formulations such as self-emulsifying systems would be preferable for improving the oral administration of this drug. Regarding the apparent permeability, different studies show different values. In parallel artificial membrane permeability study in gastrointestinal tract model, the apparent permeability was low. In CACO2 cell study, the cells that represent the enterocytes, the permeability was reported to be slightly higher, and in the study performed with porcine esophageal mucosa that substituted for the intestinal membrane, in that study, slightly higher apparent permeability was reported. Moreover, CBD was reported to be able to accumulate in the oral mucosa and continues its release after the discontinuation of the drug formulation administration. This way, we can consider cannabidiol according to biopharmaceutical classification system class 2 drug, or in class in developability classification system class 2B drug, meaning that its absorption is mostly limited by its poor solubility. However, the PAMPA results showed that its absorption is permeability limited. In this case, we considered the daily dose of cannabidiol, the maximum, despite the products vary, being up to 700 milligrams orally. Regarding the transport, cannabidiol was not mentioned to be uptaken by any active transport, so the passive diffusion is suggested. Nevertheless, here we have encountered conflicting data in the literature on the efflux transporter effects. Cannabidiol was mentioned to be a substrate for PGP and for the breast cancer resistance protein efflux transporters. Nevertheless, some sources say that this is an inhibitor. Some sources say that cannabidiol is a substrate only. Importantly, cannabidiol is taken up into lipoproteins, into kilomicrons, providing the lymphatic absorption, and this is thought to be the primary mechanism of increased absorption in the presence of lipids for cannabidiol. In animal studies, in rat studies, the concentration of cannabidiol in the lymph was found to be up to 250 times higher than in plasma. What is more, lymphatic transport bypasses the liver, and that contributes to the bioavailability of cannabidiol, limiting its pre-systemic metabolism, but also the Tmax, the time to reach the maximum concentration in plasma via the lymphatic transport increases. Regarding the first pass metabolism, there are two major cytochrome P450 enzymes, CYP2D19 and CYP3A4, that could be found also in the gastrointestinal lining, in the enterocytes, and of course in the liver, contribute to extensive first pass metabolism. Cannabidiol is a high clearance compound, and upon the first passage through the liver, approximately 70% of the total absorbed drug is being extracted, cleared. Major primarily metabolite, 7-hydroxycannabidiol, is being formed, and of course this first pass metabolism is a big challenge for oral formulation, as it significantly limits the oral absorption of cannabidiol. A bit more about the absorption of oral bioavailability. As I have already said, the oral absorption is low, incomplete, and solubility limited. It takes place mainly in the duodenum and of course in the proximal ileum. The oral bioavailability reported is up to 19%, but it is increasing in the presence of other cannabinoids. For example, even 0.2% of non-psychoactive THC addition increases the bioavailability of oral cannabidiol by 22%, increasing it up to 40% approximately. But please mind, these are the results from the cell studies. The high fat presence in meal increases bioavailability of cannabidiol approximately fourfold, and reduces the variability in bioavailability parameters. That's why we all know that the existing formulations containing cannabidiol are also lipid-based to standardize the bioavailability among patients, more or less. Talking about other pharmacokinetic parameters such as Cmax and area under the concentration time curve increases approximately 4 times in fed state compared to the fasted state. Distribution: the cannabidiol is very extensively plasma protein bound, more than 90%. It has high volume of distribution, approximately 33 liters per kilogram, and in chronic administration, this highly lipophilic substance results in accumulation. No surprise, the high lipophilicity also dictates very good permeation across the blood-brain barrier and the distribution into peripheral compartments. We have already heard very good explanation about different receptors where cannabidiol can affect. Here we have some of them mentioned in the CNS, in the muscle, in the adipose tissue, and so on. Regarding the metabolism of cannabidiol, the primary site of metabolism of this drug is, of course, the liver. In patients with hepatic impairment, the systemic exposure to cannabidiol increases approximately 2 to 5-fold. The phase 1 metabolism is mostly associated with cytochrome P450 enzyme mediated hydroxylation, where already mentioned CYP3A4 and CYP2D19 play major role. Other enzymes might also be involved. Second phase is the glucuronidation of the parent compound cannabidiol and formed metabolites. Metabolites are identified more than 100. This drug is very likely to be hydroxylated, but the major metabolites are 2, the 7-hydroxy cannabidiol, the one that is active and that possesses anticonvulsant activities similar to those of the parent compound. Another, the major circulating metabolite is inactive, is a carboxylated form of cannabidiol, the metabolite which is a product of further metabolism of 7-hydroxycannabidiol, 7 that's another metabolite. Pardon for my brain freeze. Nevertheless, there are more than 100 metabolites identified. Major route of excretion most likely is the kidney in the glucuronidated form. The clearance is approximately 74 liters per hour, but apparent clearance, depending on different oral dosage forms, depending on different absorption, was reported to be very highly variable, starting from 500 to more than 4,000 liters per hour, and that's associated with extensive metabolism of this drug. Half-life, again, is highly variable. Some sources mention 1 hour, some sources mention up to 70 hours, and that's also related to the frequency of drug administration, single versus multiple, continuous chronic exposure to the drug. It is related to the fasted or fed state. Approximately 5% or even less of absorbed cannabidiol is excreted and changed in urine, but of course, mostly the conjugates of the parent compound and conjugates of metabolites are excreted. More than 80% of orally taken CBD and metabolites are excreted in feces. Cannabidiol and its metabolites were identified in bile, but here we also have identified the controversy in the published literature. Some early studies reported that the major excretory product of cannabidiol in humans is unchanged CBD. However, we know how many and how different metabolites there are. We suggest that this controversy might be due to the incompleteness of the analytical methods used in earlier studies. To summarize, cannabidiol is chemically unstable, especially in acidic environment, and that's critical for dosage form formulation. It undergoes extensive metabolism. No surprise, it has low oral bioavailability. We didn't find enough information on pure CBD biopharmaceutical properties such as solubility and permeability, because most of the studies deal with already improved formulations such as nanoemulsions, self-emulsifying systems containing cannabidiol, but not pure cannabidiol substance itself. We have found some conflicting information on the literature regarding the excretion, unchanged versus metabolized, and regarding the transporters, the PGP and the BCRP. It is important to highlight that the lymphatic transport plays an important role in cannabidiol delivery to systemic circulation. And what we found especially interesting is that degradation products or metabolism products, we can call them differently, affect the transport and the absorption, and the already mentioned CBD tends to inhibit the intestinal membrane CBD efflux, thus increasing the bioavailability of cannabidiol. We suggest that the combination of the two substances actually does better to promote the higher bioavailability of cannabidiol from oral routes of administration. Strategies for the formulation. First of all, considering that this drug can be classified as with the biopharmaceutical classification system as a class 2 drug with permeation difficulties, the drug can be solubilized at the site of absorption, and that can be achieved with amorphization, solvent-based impregnation, freeze drying or spray drying techniques, complexation with cyclodextrins, or encapsulation with polymeric hydrophilic polymers. If we consider that cannabidiol is class 4 BSC drug, then to overcome the problems with its poor water solubility, different lipidic formulations can be incorporated. That's what exactly happening. Incorporation of CBD into the lipid-based formulation, such as self-emulsifying systems with fatty oils, lipid surfactants and co-solvents, use of fatty acid containing eutectic systems, nanoformulations, or different liposomal formulation. The good point about them that liposomal formulations not only might improve the solubility of cannabidiol to improve its deliver into the systemic circulation, but also would improve the formulation stability. I will repeat what is very well known, but co-administration of cannabidiol with foods and oils is currently the most effective strategy for enhancing the oral bioavailability and reducing the absorption variability of cannabidiol. Well, our work is now published, and you are welcome to have a look at the article for more details. And that actually concludes my presentation. Thank you very much for your attention, and I would be happy to answer your questions. Thank you.
Thank you very much for a great talk, Andrei. In fact, you just answered one of the questions that came up in the Q&A from Professor Trevor Jones, who's chair of the UK Cannabinoid R&D group, who said, is this going to be published as a paper? But it is already published. So if you could perhaps maybe at some point in the Q&A box, if there's a link to this, that would be very helpful for our viewers, I think. There were a couple of other questions that came up, which I'm trying to find now. It looks like it's moved. Around the entero, it's disappeared. Whilst I'm looking for it, I have a question. A lot of what you've talked about was looked at following a single acute dose administration of CBD, and as you'll have heard from Murray's talk previously, the trial is going to be looking at chronic administration. How does chronic administration of CBD compare to what you see after just a single dose, in terms of, can you reach a steady state level? You mentioned that it's stored in some tissues. My understanding is that, for example, adipose tissue can act as a storage depot for CBD. So if you could just maybe expand on the difference between acute and chronic, that would be helpful.
Well, according to what we have found, the single administration with so high extraction in the liver is not going to do a lot, but repeated administration, continuous administration results in the accumulation of the substance, and that, I believe, is more associated with the pharmacological effect. So upon single administration, most of the drug will be very quickly and very actively cleared.
Okay.
And I think, Cherry, you'll see the question about enterohepatic circulation.
Yes, I found it now.
I think the speaker's already answered that directly online. So.
Oh, okay, all right. That's why it disappeared off my screen. Are there any other questions pending being asked then? Okay.
Regarding your question, I have already shared the DOI of our paper to.
Yes, I see that. Yeah, no, I don't. Clive, I don't think that's the answer to the question from Bo Sals, a PhD candidate in the Netherlands, where it said, you mentioned lymphatic absorption as a possible explanation for increased bioavailability of CBD in the presence of lipids after a high-fat meal intake. Do you think enterohepatic recirculation could also play a role?
It could, but I suggest that, considering so high degree of metabolism, not much of the pure CBD. Interestingly, we have also, and we talk about it in our paper, the glucuronides, the secondary metabolism products may be broken back into the active substance, so that might result in the secondary absorption. But the amount of information on this, the amount of very basic information about this substance, cannabidiol, is very limited. Everyone is focusing already on advanced formulations, but let's say the basic information is lacking, but luckily there is a lot of things to study ahead.
Could I ask a question, if you don't mind? You mentioned several times, Dr. Sitov, in your presentation, that the presence of even low levels of THC enhance the bioavailability of CBD. And I wanted to understand a bit more why you think that happens.
I consider that might be associated with the competition at the same efflux transporters. One can be blocking the efflux of cannabidiol. THC might be blocking the efflux, so that would reduce it that way. This is what came to my mind when I was reading the cited publication.
And I suppose coming back to the question I put to Professor Fallon earlier, the question that came up earlier about CBD versus the whole smoking the cannabis plant, where you've got THC and many other cannabinoids, is whether or not by just using pure CBD, particularly if the formulation is not correct, you'll end up not necessarily getting an optimal biological effect. And I just wonder what you thought about that.
It's difficult for me to say what is better. If I understood your question correctly, what is better, the cannabidiol itself or the composite, the whole complex, considering that after the administration, including the oral administration, cannabidiol will be converted into all other things. You administer cannabidiol or you administer cannabidiol in combination, you end up with having a combination of different substances because already cyclization back into the THC can happen in the gastrointestinal tract. At how high degree, at what amount, that's another question. But after taking cannabidiol, patients will receive many different molecules.
Yeah, and I was interested because obviously the stomach is acidic and you can say you can get cyclization there, but also most inflammatory lesions, coming back to some of the earlier discussion we've had about the effect of CBD on inflammatory cells, are also about pH 5.5. So they are acidic, and one wonders whether or not you're getting conversion to THC locally in the tissue.
That would be a very interesting thing to study. I haven't encountered anything reporting that.
Yeah,
it sounds like a great
can I just pop in with
plan for the study, please?
Yeah, in terms of things like pH, and you talked at the beginning about the stability of CBD, and okay, it's relatively stable up to a fairly high temperature. I think it was up to about 60 degrees, but you know, with any preparation, storage is incredibly important, and I just wonder whether you know, the commercially available CBD products that people can buy are reported to be extremely variable in the CBD content that they have. In fact, in some cases, there's little or none in there.
I think it's a well-known
could
fact.
that be one of the reasons, is that inappropriate storage and management, or just that there's not any there in the first instance?
Absolutely, yes. We have already mentioned that the drug is sensitive to light, how frequently you open the container, whether it's a oil solution or box of gummies. It is sensitive to oxygenation. So the contact with oxygen might play a role, heat altogether, and with let's say prescription drugs, the different food supplements, the variability of the actual content of cannabidiol is, I can't explain it. It can be everything, it can be nothing inside.
Well, I think I've just looked at, there's a comment from Professor David Cahoon from UCL on the chat about he's not convinced about the evidence of the usefulness of CBD to be very unconvincing. And I think, as Katie said at the beginning, and I think he's quite clear, we have a lot of preclinical evidence for what CBD can do, the interactions it has with different receptors, but the data for clinical use in proper controlled trials, I think, is very limited. And hence really why Mary, and there are other trials being planned as well with a standardized formulation that is well validated, well characterized, is exactly why we need to do these trials, because at the moment I think the clinical data is unconvincing because of the lack of good evidence.
I think maybe this is a good opportunity to open this up to invite the other speakers to join us. Clive, I think.
Yeah, I think that'd be good. So we've got time for some further questions, more general questions that have come up during the afternoon, or others that you may have for the speakers. I think Katie, unfortunately, being in Australia, has probably already gone to sleep and not available. But if there are questions to her, please let
No,
us
she's
know.
here. She's here, wide
Right.
awake.
Okay, Katie. Well, so there is one question that came up earlier that we didn't ask you, Katie, that has come up from Dr. Guillain Vordan, asking about, is there a differential effect of THC and CBD on different types of cancer cells? And maybe we could ask Mary as well for this. Is it known whether or not CBD has any interaction with existing chemotherapies? So firstly, Katie, and then maybe Mary, if there's anything you can add to that, or if there's any data known about it.
I don't have very much on it, but I do know that it does vary depending on definitely depending on the cancer. But from my knowledge, the CBD enriched versions tend to be better at killing the cancer cells than the THC.
Okay. And then what about interaction, Mary? Do you know, I know you were talking in your trials about people who've had chemotherapy and have stopped successfully, and more to do with the post-chemotherapy neuropathic pain. But is there anything known or that you've come across people taking CBD with interacting with cancer chemotherapy?
I think that's a really important question, because there have been a number of attempts to attenuate neurotoxicity, during the administration of chemotherapy. It's obviously a big pharma issue. And the drugs that tend to be effective attenuate the tumoricidal effect, whereas there's no evidence that CBD causes attenuation of tumoricidal effect at all. And of course, there are studies in breast cancer and brain cancer to the contrary to show, preclinical work showing tumoricidal effects per se. So at present, with the way knowledge is, we feel safe there.
Okay. So another question that came up earlier, and this may be a game one for you, Katie, is that you described CBD, there's literature showing it has an antioxidant effect. And the question from Abimbola Suumni is whether that could explain its antidepressant activity that's been reported, and whether or not the specific molecular target for that antioxidant antidepressant effect is known, and whether that is any different or beneficial over existing antidepressant classes of drug. So Katie, I don't know if you've got any evidence, or if there is any evidence out there.
I'd have to say that it does work on the serotonin receptor as an antidepressant. But I also think, given that it works on so many molecules, it's not just its antioxidant behavior that is actually benefiting having antidepressant activity. So it has a far broader scope than that, and therefore you might be getting effects on there's some epigenetic mechanisms that it works on as well, and I don't know a great deal about them, but it can affect DNA methylation and histone modification, other things like that. But it also can affect other factors that affect depression, and in so doing, like anxiety and other comorbidities, and in so doing, it has a broader scope and therefore a more holistic impact as an antidepressant rather than the current antidepressants.
So to Professor Cahoon's point earlier about he's not convinced that there's some good evidence for CBD having any benefit. Particularly, we didn't really cover the evidence around the claims that it might have antidepressant anxiolytic activities. So are there good controlled randomized clinical trials of looking at CBD in the treatment of depression, and how does that stack up if they exist against existing antidepressant classes?
In most cases, there is a secondary outcome if there are trials, and I'd have to say again, the consistency in the type and administration of the CBD given is not there. So yeah. And the studies that do look at it, even though there is an effect, it tends to be as a secondary outcome.
Okay. So again, there's a clear need to do proper placebo, randomized controlled trials with validated, well-characterized formulations.
Yes.
I mean, I think that's come through loud and clear all afternoon. So are there any other questions?
Question here, Clive, that
Yes.
I think was addressed to Oliver from a PhD student at UCL School of Pharmacy, and was asking about whether the observed effect of ACBA on 5-lipoxygenase was exclusive to that boswellic acid, or is it also observed on other boswellic acids? And I think they're interested because they've seen companies combining boswellia extracts with cannabinoid extracts.
Yeah, I mean, this is a question related to not to CBD, but to ACBA, and I can clearly answer. We need the keto moiety in these boswellic extracts of these boswellic acids for the activity. So the same molecule without the keto moiety, which is the acetyl boswellic acid, is not active in this respect. So there are clear structure-activity relationships. For CBD, as you have seen, most of the cannabinoids were active against 5-lipoxygenase and also apparently to 15-LOX. So there are not so clear structure-activity relationships for cannabinoids in this respect.
Yeah. So maybe while we've got you, actually, Oliver, I'd like to ask you a question as well. I mean, a number of groups have shown preclinically that if you give CBD, you can have an effect on inflammatory cell influx into various tissues, which is a hallmark of a number of inflammatory diseases. And I wondered, through the work that you've done looking at the ability of CBD to affect lipid metabolism in cells, have you ever looked at whether that alters the expression of adhesion molecules on the surface of the endothelium or leukocytes that are critical for that cell trafficking? Because that may explain why you get a reduction in the recruitment in an acute setting, a recruitment of inflammatory cells into the tissue.
No, we have not looked at adhesion molecule expression, but our hypothesis is that since these cannabinoids, in particular CBD, represses the formation of leukotriene B4, which is a very potent chemotactic and chemokinetic agent, this probably is the mechanism of how the cell infiltration, in particular of neutrophils or monocytes, could be repressed. So by repressing the biosynthesis of the chemotactic agent, so to say.
Yeah, although acutely, of course, both the 5 heats and the 15 heats, which are the other side of your equation, are both chemoattractant for neutrophils. So I think there's this sort of acute versus chronic, which you made very clear that a lot of our experiments look at acute inflammation and we don't really look at the resolution.
Yes.
And I think when you look chronically, as you say, the pattern of lipoxygenase metabolites changes, and therefore the overall impact on how long those cells may exist in a tissue, for example, I think becomes then critical.
Absolutely. So what we have done is only one in vivo experiment where we studied the activities over 4 hours, which is of course an acute inflammation. So we have not yet tested these settings, so to say, in a more chronic model where like in a colitis model where the mice are observed over 7 or 40 days. I think that would be urgently required to do. Definitely.
Okay. Good. Well, there's one other comment come up from Professor Cahoon, who says he's surprised to hear people still talking about antioxidants as though they were beneficial, and has put a link in which people might be interested to see. So thank you, David, for sharing that with us. And I'm not sure, Cherry, whether you have any other questions. There's not any other ones coming up on here. But unless there are any other questions, I think we're...
There's one just come up from Andrew Brown, Clive. He'd actually put it in the chat and I asked him to post it into the Q&A. He sent a link to a website where, and I think this might be something that Andres can help with. It's got a cannabidiol derivative, which is reported to be more bioavailable. but it has a different structure. So it would be good to see some independent studies of its broader pharmacological profile. I don't know if you've come across this at all, Andre?
Definitely, some substances might be much more stable than cannabidiol and have much better pharmacokinetic profiles. So unfortunately, I'm not really aware of this particular substance, as chemically there is possible to synthesize many different molecules. But one final remark that I wanted to contribute also, I have mentioned quite a lot about the instability of cannabidiol substance itself and its metabolism and so on. We shouldn't forget very high inter-individual and even intra-individual variability in absorption and metabolism of this drug. That's why finding the perfect dose and the perfect formulation would be a very tough challenge for my colleagues, formulation scientists, and the clinicians.
Well, I think on that very challenging point, Dr. Sitovas, I think we'll probably round up the session, and it really just leaves me to thank all of the speakers. I think we've had an interesting afternoon. To thank you, Cherry, for putting this program together and setting the meeting up. Also to our sponsors this afternoon at Ananda Developments, particularly Melissa Sturgis and Charlie Morgan. I think it's highlighted a lot of the challenges around this. I think as Katie put at the beginning, there's a huge amount of preclinical data, cellular data, but there's a lot less convincing data from randomized placebo-controlled trials of the usefulness of CBD. And we hope that some of the trials like yours, Mary, will help answer some of those questions, because I think if it translates into clinical practice, it could be something useful, but at the moment a lot of that data is lacking. I'd also like to thank the BPS for agreeing to host this meeting, and particularly to Bonnie and Isabel and Nicola in the events team, and also to all the attendees. We've had a good attendance all afternoon, and for your questions. And just on the screen now, you'll see that there are the number of upcoming events that the BPS runs, a number of events across the year. Some of them are listed here, and you can always go to the BPS website to find out more. And for those of you interested, our annual meeting is going to be in Harrogate in December, and it's not too late to register, even though you've missed the abstract deadline. And we hope to see more of you at the meeting. It's a very broad church of people who go to the meeting and the type of pharmacology that's discussed. So with that, I thank you again, Cherry, for organising such a good event, and the speakers, and to everybody for their questions. And I wish you all a good evening, and for you, Katie, a good sleep. And also from our colleagues from abroad, particularly in Indonesia, because you must also be about to try to go to bed, and if you're not, you should be. So with that, I thank everybody, and look forward to catching up with some of you again very soon. Thank you.
And thank you from me as well. And thank you, Clive, for your excellent co-chairing.
Thank you.
Thank you to all.
Thank you. Bye-bye.
Great
Okay.
job. Bye-bye.
Bye.
Bye.